system, and the nervous system is the one that we said this is rapid communication. One thing that we didn't really get into was especially that he wins over everything. The nervous system is always going to win over everything. The nervous system is going to be the last to go. Even after your heart has stopped, you're still going to have nervous twitching. This is where that agonal gasp comes in, because we're twitching the diaphragm. So he is the master controller. It's a very rapid communication, and it's actually electrical signals. We've talked about the electrical signals when we talked about action potential in the muscle and the change in the resting membrane potential. Now, what makes the electrical signals is chemicals. So neurotransmitters are proteins that neurons make, and they are the guys that end up creating what kind of electrical signal is going to happen. This next part sounds like a lot of words, but it's total sense. There's 3 functions. You're going to sense something, your brain's going to decide what to do with that sensation, and then it's going to act on it. So the one that we always think of is moving. So I sense that there is a little spider on my arm. My brain decides that he does not like spiders, and so you scream and swat the spider off. But sometimes it's something that you are not in charge of, because we have a nervous system that we're in charge of, and then we have a nervous system that we're not in charge of. So for example, your body recognizes, he senses that you ate some protein. So then you're going to have, with that sensation, your body is going to decide, okay, I recognize that's protein, so I'm going to put out the correct digestive enzyme to digest that protein. So you get the idea. My GI tract recognizes that I just ate some pizza, and so my stomach recognizes that and acts on it. So motor output starts churning up the pizza, and gastric juices start to come out of the gastric glands, and the small intestines start to move more to get that pizza moving down the GI tract, and so you can absorb the food. So that's a motor output, but it's not one that you're in charge of. All of those things, the secretion of the glands, the stomach churning the food, and the intestines becoming an inchworm, that's all a motor response, but you're not in charge of it. So this is showing the one that this is the one that we would relate to most of the time. You see a glass of water and you think, I'm kind of thirsty. That looks good. So your brain says, well, you should go and pick up that glass of water and put it up to your mouth and swallow. So we have 2 basic parts to the nervous system. We have the central nervous system, which is the brain and spinal cord. And then we have the peripheral nervous system. That's everything else. We have, whenever it's in the peripheral nervous system, we have lumps of axons called nerves. So we have spinal nerves that have left the spinal cord, and they're going to go out towards the periphery, so towards your body, where you get to sense those things, and then it's going to bring that information back to the spinal cord in that same segment of spinal cord. Then we have cranial nerves. So this is still peripheral, even though we call them cranial nerves. They have left the brain. They received the information, and then the information goes back to the brain, but they're still nerves. So you have spinal nerves that are branching off the spinal cord, and cranial nerves that are branching off of the brain. So, for example, we have those nerves that, let's say, maybe you've heard of somebody that has sciatica. Well, sciatica is the sciatic nerve being pinched, but it's probably being pinched pretty high up, closer to the spinal cord, where you might have some sort of back problem, maybe arthritis, maybe slipping disc, maybe just an old injury. Now the peripheral nervous system, okay, so remember cranial nervous system is brain and spinal cord. Peripheral nervous system is everything but the brain and spinal cord. So the peripheral nervous system job is to sense things. So that's afferent, arriving at the nervous system, the central nervous system, and it has an efferent, the motor division. So this is where things are leaving the central nervous system, going out into the body. A somatic sensory is going to be things that we feel. So I mentioned the spider. So we feel something like that and we consciously recognize it. Maybe you have some arthritis and you notice joint pain with barometric pressure changes. You sense that. And then, of course, skeletal muscles. You can sense that you are using them, contracting them. Maybe you exercise them. Maybe you got some lactic acid buildup and they're sore from yesterday. These kind of things, you can sense it. Visceral sensation, that would be all your guts. This is not something that you really are thinking about and you can't really tell is going on. So like your visceral sensation of eating the protein that I just mentioned. It's still sensory. It's just not something that you are aware of. It's not something that you are in charge of, but it still has to get processed to the brain so the brain can decide, okay, what am I going to do with this information? He ate some protein, so therefore I'm going to need the right hormone that's going to tell the right organ to put out the right digestive enzyme. You see? Okay, so it senses, and it's not a sensation you're in charge of. Somatic are things that you can recognize, visceral are things you can't recognize. And then we have to move things. So somatic nervous system, swat off the spider. Go for a jog. Go get yourself a Coke. Whatever. Oh, I'm tired of studying this. I'm going to go get me some coffee or some tea. Autonomic nervous system, you're not in charge of that guy. Things are going to move inside your body constantly. That's from chapter 1. So we are constantly in motion. Even when we're sleeping, there's millions upon millions upon millions of things moving inside your body, and you're not in charge of that. That would be the autonomic nervous system. So that was sensation. Now motor, I've already kind of described that. You know that you are in charge of moving things. That would be a somatic system. Autonomic nervous system, you're not in charge of moving things. Now this guy is, you've probably heard of it before. Maybe you already know what I'm fixing to say. But you have a fight or flight section of the autonomic nervous system, and you have a rest and digest section of the autonomic system. Fight or flight is sympathetic. Rest and digest is parasympathetic, and we will get into that in a different PowerPoint. This is just a summary. Okay, so I told you when we did tissues that there's neurons and there's a bunch of other cells, actually a lot of other cells. And I said we'd get into that in the nervous system. Well, here we are. They're called neuroglia. You have some in the central nervous system, and then you have some in the peripheral nervous system. These cells are supportive of the neurons that you've already learned about. So the neurons are the excitable cells that do the zapping. I need this target to do this. And the glial cells is sometimes what they're called as well. The glial cells are going to be supporting, and they're very close by, whether it's in the central nervous system or the peripheral nervous system. This slide's got the 4 from the central nervous system listed: astrocytes, microglial cells, ependymal cells, and oligodendrocytes. Okay, so the first one we're going to go over is astrocytes. And again, we're in the central nervous system. There's 4 in the central nervous system. There's 2 in the peripheral nervous system. We'll get to those in a second. Astrocytes are the most abundant glial cells in the central nervous system. They're helpers. They're almost, glial cells were thought originally to be glue between neurons. And then they've discovered that they actually have a lot of function. But if any of them is a glue, it's this guy, because he is hanging out right next to neurons. Maybe sometimes he's right between the synapses, maybe right between the capillaries. Sometimes he helps connect capillaries and pass things to other areas. Maybe that neuron needs a little extra glucose. Well, here, let me help you with that. I'm going to get some glucose out of that capillary and get it to you. If you have some young neurons and they're branching, so little children and they're learning, I've told y'all when you study, you have branching off of your neurons. So you create more and more pathways and more connections when you repetitively study. So young children, how they're learning a lot, right? They're learning to walk and talk and recognize things and learn colors and alphabet and blah, 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 blah. So those neurons, as they're creating their branches, the astrocytes help guide those branches to get to a target, another neuron usually. They just do so much stuff. If you smoke some weed, enough that you end up killing some neurons off. Usually it just pulls the dendrites away, but the dendrites are the guy receiving information. So what good is that neuron if he can't receive information, right? Anyway, or let's say a harder drug, cocaine. You kill yourself some neurons don't go through mitosis. That area is going to be replaced by astrocytes because they can go through mitosis, but they don't communicate. They don't send electrical signals that help you think. So you lost a thinking cell and you replaced it with a supportive cell that's going to make sure that whatever neurons you have left are taken care of, but they don't help you think. They won't come back. So here's a picture of an astrocyte showing he's got a connection there between a capillary and between a neuron. And he's making sure that that neuron gets all the nourishment he needs. Microglial cells are like a macrophage. So they're kind of hanging out, and hopefully most of the time they're very bored. But if you have a neuron that starts to die, or if you get some sort of pathogen that enters into the brain, this cell, the microglial cell, will go and clean up the dying cell, or hopefully engulf and kill the pathogen that's entered into the brain. Ependymal cells are super cool. They have cilia. It says maybe ciliated. I feel like I should change that. We should just recognize that they have cilia. And what they're doing is they are lining the ventricles, any of the cavities in the spinal cord that has cerebral spinal fluid. They are a connection between the capillaries and that fluid passageway, and they are genuinely the guy that is helping make cerebral spinal fluid. So he's connecting two capillaries, and he's lining those ventricles. He's lining the area that has cerebral spinal fluid. The ventricles have cerebral spinal fluid, but you also have cerebral spinal fluid in and around the spinal cord. So these ependymal cells are lining all those passageways that have cerebral spinal fluid, and he's the guy that's helping make the cerebral spinal fluid. He also helps make a barrier. If The brain needs protection. It needs a blood-brain barrier, which is a extra tight capillaries that's trying to keep pathogens out of your brain. So this is what they look like. Oops. Oh, well, you saw the picture. You can look at it again later. Okay, now some neurons are myelinated, which means they are wrapped in like a fatty sheath. Whenever they are wrapped in a fatty sheath, that's called, they're myelinated, but they process information a lot faster. So we have myelination in both the central nervous system and the peripheral nervous system. The cells that myelinate in the central nervous system are called oligodendrocytes. Now in your lab book, you're going to meet the other one that in the peripheral nervous system, and it's called a Schwann cell. We'll get there in a minute. They both wrap themselves around that axon, but in a different way. So you can see the cell body, it's round and it has a process sticking off of it. And these little processes are jelly rolling around axons. In the central nervous system, one oligodendrocyte can do many axons. So he's got cytoplasmic extensions that are going and wrapping around at different locations. Okay, so the glial cells in the peripheral nervous system, there's only two. Very similar to the astrocytes are a cell called a satellite cell. Now they're going to surround cell bodies. So you'll see in a little bit that there's really not too terribly many cell bodies in the peripheral nervous system. There's some, but the majority of the cell bodies are going to be in the central nervous system. So supportive. So making sure that that neuron gets whatever he needs. The Schwann cell is just like the oligodendrocyte. He myelinates the axons, but instead of putting out little cytoplasmic extensions like feet, and the little feet wrapping around, what this guy is going to do is his whole cell is going to wrap around a axon, usually. Always exceptions, right? So here's a picture showing how each of those little Schwann cells are jelly rolled completely around an axon. And axons are going to end up being called nerve fibers in the peripheral nervous system. So you met the muscle fiber, now this is a nerve fiber, because axons can be quite long. Okay, so here's a little Google map kind of neuron. You're fixing to learn that there's different kind of neurons. The only one I've drawn on the board for you thus far has been a multipolar neuron. But this is a unipolar neuron. And unipolar neurons, they're sensory, and they are going to be out in the periphery because that's where we sense things. So you sense something going on in the periphery of your body, whether it's your stomach contents or your muscles or your boy and girl parts or your heart. You sense things, and we've got to send that information to the central nervous system. So when we sense things, it's going to be this funny looking guy right here. This, I call him a Google map. Do you appreciate he looks like a Google map? So that cell body is wrapped with some satellite cells to make sure that that neuron's getting whatever nourishment he needs. And this particular neuron is wrapped with a whole bunch of Schwann cells, so he is myelinated, so it's going to be very fast. Myelin makes them fast. So let's go through these different neurons that we have and what exactly they are. I don't really like calling them nerve cells. Let's just stick with neurons because a cell is a neuron. A whole bunch of axons bundled together is going to be a nerve. So that's a little bit confusing if you call a neuron a nerve, because they're not the same thing. So I should go and edit this and take out that term nerve cell. But that's what everybody's heard of it as, a nerve. But it's actually a bundle of a bunch of the axons from a neuron. Okay. So they're the guy that's creating the electrical impulses thanks to some chemicals called neurotransmitters. They do not go through mitosis. So amitotic means no mitosis. They are extremely long-lived. They should last your whole lifetime, you hope. And wow, these guys need sugar and oxygen. They're only going to get their energy from glucose. No fatty acids are helping out with this. Must have glucose. If you've done the keto diet and took sugar out of your diet and you're just eating protein and fats, you probably initially were very, very tired. And that was because your brain wasn't getting enough glucose to keep those neurons firing. Probably got some headaches, probably just in general didn't feel good. And by the way, this is not a good diet. Okay, so they all have a cell body and one or more processes. The cell body is also called a soma. So sometimes I might call it a soma. And he's just like any other cell. He's got the nucleus in the middle, and he's going to have protein synthesis going on. He's got his rough ER is going to have a new name called a Nissl body. I don't know why, but it does, and that's in your lab book. So I left it in here because I'm trying to repeat. There's some things in this PowerPoint that I took out because it didn't have anything, or it wasn't precisely as the lab book portrayed it. So I'm trying to stick with lab book. But anyway, it's a cell, so he needs to make proteins. The biggest protein that guy needs is neurotransmitters, but he also needs those integral protein gates to pass that electrical conduction and to let in some calcium so he can officially let go of his neurotransmitter, just like at the neuromuscular junction. Remember, those are proteins, so you have to do protein synthesis. Let's see, he's going to have his cell membrane at the soma is going to be the area that's receiving information. So maybe somebody's axon terminal is touching the soma, or maybe touching one of his dendrites. But regardless, that's usually where that neuron is going to receive his information. Most neurons, I mentioned this earlier, most neuron cell bodies are located in the central nervous system. We are going to have some located in the periphery. That's why you saw the satellite cell. That little Google map guy, the unipolar guy, is in the periphery. But a bunch of cell bodies in the central nervous system is going to be a nucleus. Multiple nucleus is a nuclei. What was I going to say about that? Yes, this is the third nucleus that you had to learn about. So you've learned about the nucleus that's inside of a cell. You've learned about the nucleus of an atom. And now we're going to have a cluster of cell bodies in the central nervous system, and that's going to be the nuclei. In the peripheral nervous system, when you do find cell bodies, again, they're not that common, but they're going to be clustered in a little swelling called a ganglion, and multiple ganglion is a ganglia. So we have several ganglia in the body, but nothing compared to all the passageway of tiny little neuron fibers that we have. Okay, leaving the cell body, we are going to have dendrites, and we're going to have axons. So the branches that are off the cell body usually are going to be dendrites receiving information, and then you're going to have an axon that's sending information. Those areas that have these processes in the central nervous system, so not the cell body, in the central nervous system, a group of cell bodies is called a nucleus, but the processes that are coming off, the axons especially, are going to be tracts. So a bundle of axons in the central nervous system is going to be called a tract. A bundle of axons in the peripheral nervous system is going to be called nerves. Now, sometimes the dendrite's included in that. That's splitting hairs. It's not a cell body. So if you have a nerve, it's processes. If you have a tract, it's processes. Most of the time, the processes are axons. So this is the multipolar one that I've drawn for you several times. The dendrites are little branchings from the cell body that are receiving information, and the axons are usually quite long, and they are sending information. This one has, that's labeling the myelin wraps as Schwann cells, so therefore this is a peripheral neuron, not a central nervous system. Okay, so again, dendrites are going to have information that they're sending to the cell body. Now, each little signal that he's sending is called a graded potential, and we kind of touched a little bit on graded potentials in muscle, but not deeply. But here we go. I told you if you had a zap to the muscle, but it just changed the charge of the resting membrane potential from -70 to -68, that's not going to be enough to make that muscle contract. It has to hit a threshold number. This is exactly the same with the neurons. So all these dendrites are receiving little bitty zaps and sending these little bitty zaps to the neuron. If you have enough of these dendrites or enough repetitive stimulation to one dendrite to change the cell membrane to threshold, then that axon is going to fire. But because each of these guys might be receiving just tiny little ones, each little one is going to have to add up to a big enough change for the axon to fire. So those little messages come in from each little dendrite are going to be called graded potential, and they're usually from a very short distance because dendrites are usually smaller, shorter. Okay, the axon. So neurons usually have one axon, and that immediately where the axon is leaving the cell body, the soma, is going to be called an axon hillock. Note, this is in your lab book. Why is that hillock important? It's loaded with gates. It's loaded with those little integral proteins that's going to help things come in and out of the cell. Okay, then as it keeps moving, so initially it's a hillock, and as it keeps going, it gets a little bit skinnier, and this is going to actually be the fibers. So the axon often will be called a nerve fiber. You put a whole bunch of nerve fibers together, and we're going to bundle them together, and it's going to look just like the muscle when you cut it. There's going to be fascicles, and there's going to be tiny little axons in each fascicle, and then we're going to bundle all those together to be one nerve, and they each have their own connective tissue layering, just like muscle, but we'll get there later. Axons have occasional branches called axon collaterals. This is the branching that I was kind of talking about, but they actually can create new passageways. So I'm going to learn this information, and I can send that output. Oh, I understand what that means. That means this through many, many different pathways. The more you study, the more collaterals you have. You can retrieve that information better if there's interconnections. Each axon terminal can branch out in tiny little passageways. So it can have 5 targets, it can have 10,000 targets. It's that one axon, and he splays out at the very end. And the swelling at the very end of each of those branches is called an axon terminal. And you can also call it a synaptic knob or synaptic bulb. Okay, that axon is going to be a guy that's sending the action potential. We're going to have the nerve impulses traveling down the cell membrane, just like with muscle, but instead of it be calling cell membrane, I see it. I see a typo. It should be Neurolemma, Neurillemma, not Neurolemma, but that's the cell membrane. So you already met sarcolemma, now we're going to have Neurolemma. The cell body, the soma, is the guy that's making the neurotransmitters. And the Golgi apparatus is going to package them up into little vesicles, and they can slowly travel down the axon, or quickly, depending on what's going on. Those neurotransmitters are going to go to the very ends of the axon, so to the little axon terminals, and released by exocytosis. Now, the neurotransmitter that you met, acetylcholine, is an excitatory neurotransmitter. It made that muscle zap. But you've probably heard of all kinds of different neurotransmitters, and some of them actually inhibit. So things that suppress pain inhibit pain because of the kind of neurotransmitter that's being released. I don't want to zap this nerve. I want to make it harder to zap this nerve. So instead of hanging out at negative, just since we've done negative 70 already, instead of being negative 70, maybe what I end up doing is instead of throwing a sodium with a plus charge in there, I'm going to throw some chloride in there and make that negative 70 even more negative, so it's harder to zap that nerve and make it release a painful signal. Every axon can actually carry on lots of different conversations with different neurons. So he can splay out, his little axon terminal can splay out and send his signal to multiple other neurons. If you cut an axon, it's going to die. Depending on where you cut that axon determines if that axon can regrow. Earlier, I think I failed to mention on the Schwann cell, if you're in the periphery, you're myelinated with Schwann cells. If you cut the axon fairly far away from that cell body, the Schwann cell can help that axon regrow. Now, we didn't kill the neuron. If you cut too close to the cell body, then you lose too much cytoplasm from the soma, from the cell body, and that neuron's going to die. But remember, these are just little appendages that are going pretty far out. So if you cut them, they're going to decay. But the Schwann cell, if you're in the peripheral nervous system, he can help make it come back. Okay, so I mentioned this already. He's the transport system. There's a whole bunch of cytoskeleton passing through him, and there's all kinds of things going up and down that cytoskeleton. Most of the time, it goes from the cell body towards the end. So let's say a neurotransmitter starts at this cell body and moves down towards the axon terminal. That's called anterograde. But sometimes things move toward the cell body and that would be called retrograde. Maybe we've got some organelles that are not feeling good, and they're feeling that, oh my gosh, I think I'm on my way out. Well, I'm going to go back to the cell body and find me a lysozyme to go ahead and gobble me up and recycle all my parts. Sometimes signal molecules will go up that way, but I highlighted viruses and bacterial toxins because this is how, like let's say rabies and some of the clostridial bacteria is like tetanus and botulism, they go retrograde up this axon towards the cell body to commit their crimes. Why did I leave this in here? I knew I was going to say that about polio, rabies. Rabies is fatal, by the way. Almost always. There's 2 cases maybe of people that have lived through rabies. Oh, I know why. So since they have figured out that some things can go retrograde up into the cell, and they know up into the cell body is where the DNA is, sometimes they will modify a virus. So they're using that virus's ability to get to the cell, but they have taken away the ability for that virus to hurt you, to help correct a bad gene. So if you're born with some sort of genetic problem, they're working on trying to take advantage of that ability to go retrograde back up to the soma and see if we can try and repair some of the DNA. So that's pretty cool. Probably quite difficult, but quite cool. Okay. So, myelination in the peripheral nervous system. So I mentioned this already, that the Schwann cells are going to wrap in a jelly roll fashion. And one cell is going to create one segment of myelination. From your lab book, now your lecture book gets into a little bit more details. I think I left the picture in to show you the detail, but let's just stick with everywhere that is the exterior of the cell of the neuron is going to be called neurolemma. So let's just stick with that. That's what your lab book says, and so we'll just stick with neurolemma is the cell membrane. So I mentioned that whenever something is myelinated, that action potential travels so much faster. And so why is that? Well, because the cell membranes of the Schwann cell don't have carriers or channels, so they're not letting proteins in, they're not letting ions in or out, and so they're genuinely just like a thick blanket around a pipe at when it's really cold outside. He is making sure that all the energy is staying inside that cell, and later I'll show you what else he does. So this is showing a Schwann cell. He's wrapping in a jelly roll fashion. He gets tighter and tighter and tighter, and he's genuinely insulating that that axon. Okay, so this is what your lecture book says, and this is the part that I'll show you the picture because it's awesome, but I want you to understand. We're just going to call the outside of every neuron neurolemma. There's an area, so this is from lecture. Don't worry about this for your test, but I wanted you to see this picture right here. So here's an axon right here, and here, right here, is all that myelin. Myelin's a lot of fat from the Schwann cell. So look at that jelly roll. He rolled around, I could probably count this, but I'm going to say 20 times that one Schwann cell wrapped around. Well, that squeezes his nucleus towards the outside, okay? So it makes makes the nucleus of the Schwann cell bulge, like on that previous picture, and I can't go back or else I'll lose my recording. But so your lecture book calls it the outer collar of the perinuclear cytoplasm of the Schwann cell. Let's not worry about that. Let's just call it all Neurolemma. Okay. Between these areas that have myelination, so we have a Schwann cell space, Schwann cell space, Schwann cell space. Those little spaces are called nodes of Ranvier. That's how it's written in your lab book. Your lecture book says myelin sheath gaps, formerly known as nodes of Ranvier. Your lab book says nodes of Ranvier. Let's just stick with the nodes of Ranvier. So that's one less thing for you to remember. Okay. Now some nerve fibers are slower because they're not myelinated. And does that mean that they don't have any Schwann cells? Not exactly. It means that we might have a whole bundle of neurons and one Schwann cell is wrapping around them once, but not doing that jelly roll fashion. So they definitely are thinner, they're slower, and they don't look as white. It's usually going to be free nerve endings and not something that's obvious with your eyeballs. So when you're doing gross anatomy, it's very hard to find these little tiny nerve fibers that aren't myelinated. And there's that picture again. In the central nervous system, again, we've got those oligodendrocytes. He's going to use little cytoplasmic extensions, and he's going to use his feet to wrap around pieces of a neuron. So he can actually get a whole bunch of one oligodendrocyte can do many axons. Schwann cell, his whole life, he's just wrapped around. That's a long life to just be wrapped around an axon. That's all he does, that one cell. In the central nervous system, these oligodendrocytes can make many, many, many feet and wrap around many, many, many axons in one spot. Then we're going to have more than just one oligodendrocyte doing that, so you're still going to have those little gaps, those little nodes of Ranvier. So when you look at a brain, and this is also in your lab book, you're going to see white matter and gray matter. And I'm going to tell you, on a fresh brain, white isn't exactly white. It's kind of like a light pink, but it's clearly not gray. It looks totally different. So if it's white matter, that's going to be myelinated. If it's gray matter, then it's going to be non-myelinated, and it's going to be areas of cell bodies. So remember, bundles of cell bodies in the brain is going to be called nuclei. Bundles of cell bodies in the periphery are called ganglia. But gray matter is going to be unmyelinated fibers and nuclei. This is showing how those oligodendrocytes wrap again. This was also on a previous picture. Okay, so a little bit more into the structural classification of a neuron. Multipolar is the guy that you've already met. You got a cell body, you've got an axon, and you have dendrites, usually multiple, at the soma. Multipolar is definitely the most common neuron that we have. Also in lab, the only model that you'll see is a multipolar neuron. So on a lab test, if one of the questions is a picture of a neuron, you'll see that cell body and it's receiving some dendrites, but he's got that axon that's going off with some Schwann cells. That's a multipolar neuron. Bipolar neurons only have two processes coming off of the cell body. So one's an axon, one's a dendrite, and they're very rare. You're going to find those in special senses. I try and point them out whenever I see them on a picture, but they're rare. So don't ever answer bipolar if you're not talking about smell or sight or hearing. It needs to be a special sense. And otherwise, it's either going to be multipolar or unipolar. Unipolar only has one process coming off of the cell body. And you saw him, right? The Google map. So that little upside down teardrop, something coming off of the bottom of the teardrop, that was the process. And then he's going to have that extended process. One side's receiving information, so that would be a dendrite, and the other side would be sending information. That would be his axon. Okay, so here's the structural classification. So multipolar, bipolar, unipolar, pictures. Multipolar means more than two processes off of the cell body. Bipolar means two processes off of the cell body. Unipolar means one process off of the cell body. So bipolar is the one in the middle. It's going to be like sight and smell and hearing. And you can appreciate this guy in the middle. He's got two things coming off of his cell body. Let's go to the right. Unipolar. Google map. Little Google map at the very bottom of the upside down teardrop is one extension. And then the processes off of these. Those guys are, from that extension, we're going to have branchings from that extension. And then all the way on the left, this is the one that you've met several times already. So the multipolar neuron, the cell body has many, many dendrites sticking off of it and one axon sticking off of it. But you can appreciate it has more than two things sticking off of that cell body. So multipolar. We even have other variations. Please don't memorize this. Sometimes they get named kind of like by their shape or how somebody or who discovered them. Sometimes they get named by what they do. So olfactory smell, olfaction is olfactory cell would be what helps you smell. So it's a bipolar neuron and he's getting a special name. But again, let's not memorize this. It's just pointing out that sometimes you might hear the term pyramidal cell, and this would be in much more advanced education and beyond the scope of this class. Okay, now, so that was structural classification. This is functional classification. It's very simple. I've already even mentioned two of them. Sensory and motor. You sense something, or do you help move something? Whether you're in charge of moving it or not. So if it's a somatic move, that would be like skeletal muscle, but it could be viscera moving. So you're telling your gut to be the inchworm, or tell your uterus to contract, get the baby out. Tell your pancreas to squeeze out some hormones, whatever. You're not in charge of that. Okay, so we've already been through that. Sensory, I should even take out that almost all are unipolar. That's just too confusing. If you can associate the Google map with sensation, that would be great. His cell bodies are going to be in the peripheral nervous system, in a ganglia. Motor, again, most cell bodies are in the central nervous system. Motor is going to be multipolar, and these guys are going to be carrying information to motor output in the body. There's one thing I forgot to mention. Bipolar, they're very, very rare, and I told you that they were special senses, so like sight, smell, hearing. Notice I use the word senses. So this is why that sensory is almost all are unipolar. Well, that's because bipolar is rare. So maybe a better way to write this would be almost all are unipolar except special senses, which are bipolar. Okay. Then motor is multipolar, and then the third one that I haven't mentioned yet, but now I will, is interneurons. So there's going to be, as we get into we sense something and bring that information to the central nervous system, that one guy is going to meet up with somebody else. He is going to be shaped like a multipolar neuron, and he's going to bring information up the brain, and then we're going to meet whatever in the brain and come back down and synapse with another. So you can appreciate it's just not these two guys, the one sensory neuron and the one motor neuron. Once you get into the spinal cord, we're going to have these interneurons, and they're also called association neurons. And they're like continuing the pathway. So they are maybe going to take that piece of information, that interneuron is going to go and tell the motor part of your brain, hey, I need you to send in information that there's a spider on her arm and she needs to swat it off. But then there might be another part of the pathway, another association neuron, that he sends information to your limbic system that says, hey, I don't know if you remember this or not, but you're really scared of spiders. So this would be a good time to go ahead and motor output a scream. And maybe it's going to pass another way that, let's say that instead of spiders, it's maggots. So then we might have another interneuron that's connected to this pathway, and he's going to send information to the base of your brain in the maybe in your brainstem that says, you really don't like the smell of maggots? You can go ahead and vomit now. So not only did you scream because the maggots scared you and you ran away from the maggots because you didn't want to be next to the maggots, but the third motor output was because you vomited. Because if you've ever smelled maggots before eating flesh, it stinks. So it makes you want to puke. So you get the idea. These are the sensation can be brought to different parts of the brain by the interneurons, and motor output isn't just one thing. It can be many different things because these interneurons create additional pathways once you get to the central nervous system. They're always multipolar, by the way. These guys are always multipolar. So highlight that and put that somewhere. I don't see that on here. This is kind of showing a little bit of a summary, but also I see some things on here that we haven't touched on yet, and we'll touch on in the next PowerPoint or two about how we have sensation going to the spinal cord, motor output going to a muscle. You've actually already kind of met that, but please note where that cell body is. Later when we get into the autonomic nervous system, I'll tell you about, I told you ganglia is rare in the peripheral nervous system, but wherever you have that Google map guy, that's going to be in a ganglia. We are going to see in the autonomic nervous system another ganglia, but again, most cell bodies are going to be in the central nervous system. Okay.